Hi, everyone. Thank you for joining us for the HPI 101 series, both on Zoom and in person. We're really happy to see you. So today's topic is imprinted genes in neuroscience, and we're delighted to welcome Dr. Amanda Whipple, who works in she's an assistant professor in the MCB Molecular and Cellular Bio Department over in Cambridge. She did her undergraduate work studying biochemistry at the University of Oklahoma. Then graduate work at Baylor College of Medicine, where she studied RNA binding, an RNA binding protein that was important in neuromuscular disorder. And that got her interested in RNA therapeutics. She then did an industry postdoc at Ionis Pharmaceuticals, and she started working on Angelman syndrome, which I think she'll tell you a little bit about today, which involves an imprinted gene, which is expressed, differently between the mom's copy versus dad's copy of the same gene. So she became interested in that topic, then did a postdoc at MIT, and now started her own lab very recently in 2019. Thank you, Dr. Whipple, for joining us. In terms of the format of the series, she's going to give a broad overview talk and also share some of her research in this area. If you're joining on Zoom, we invite you to type questions in the chat at any moment. And if you're joining in person, you can ask your questions at the end of the talk. I do want to remember just to say that in order for the questions to be heard on Zoom, we ask you to speak them into a mic. So one of us will run the mic over to you. And for people on Zoom, you can also ask your question by raising your hand in addition to the chat. So with that, I think we'll get started.
Great. So thanks everyone for joining me today. I've really been looking forward to this. My plan for our time together is to give you a little bit of a historical perspective on genomic imprinting, how it was discovered, some of the early and key findings of genomic imprinting. And then I'll talk briefly about some modern approaches that are being used now to further investigate genomic imprinting and its effects, and a little bit about the questions that my lab is asking. And then I'll conclude by talking in more detail about Angelman syndrome and some work that I've been involved in to develop a new therapeutic approach for treating this imprinting disorder. I'll just echo what was already said, and I would love for this to be as much of a discussion as it is a lecture. So please feel free at any time to interrupt throughout the presentation, whether or not you're in person or on Zoom. There's nothing in particular that I need to get through, and I would rather everybody leave having learned something than for me to just make it through the slides. All right, so for the presentation today, it's going to be really important to always keep in mind that mammalian cells have 2 copies of every gene. Now, if you're a scientist at the bench, you likely rarely think about this fact. And if you were to assay for the expression of your favorite gene of interest, you probably assume that the 2 gene copies within the cell are the same and are behaving the same. But in some instances, it's actually important to remember that one set of chromosomes is inherited from one's mother, and one set of chromosomes is inherited from the father. And in the early 1980s, it was actually being questioned if this matters. Does it matter that we receive genetic information from both of our parents? Or rather, is it simply sufficient to have 2 copies of every gene, independent of their parent of origin? And this question was tested in a type of experimental paradigm called a pronuclear transfer experiment, where some rather peculiar and unexpected observations were made. So in these experiments, immediately following fertilization, the 2 parental pronuclei are originally present separately within this newly fertilized egg, the single cell zygote. Those 2 nuclei then fuse together such that the single nucleus now contains the parental genetic material from both parents. And every time the cell is going to divide and replicate the DNA, it very faithfully passes that on throughout development. So you can do a type of experimental manipulation in which you actually remove the paternal pronucleus from this single cell zygote and now replace it with another maternal pronucleus. So you've now generated a zygote, which has the correct amount of genetic material. It has 2 copies of every gene, but is exclusively of a maternal origin. And it turns out that these embryos will fail to undergo proper development. It is not viable. And this is due to many things, but in particular due to a failure of the proper development of the extra embryonic tissues. You can do a very similar manipulation where you now create single cell zygotes that have two paternal genomes, this will also fail to undergo proper development, primarily due to defects in the embryonic tissues themselves. So these results were quite surprising at the time, maybe not as surprising to us now if we've already known a little bit about genomic imprinting. But it led to the hypothesis by Azim Sarani's group that perhaps a type of imprinting occurs of the parental genomes such that both parental genomes are now required for development. And I think for us here today, there's two main takeaways from these experiments. The first is that while we know just at a pure DNA sequence level, the parental genomes are genetically equivalent, they are not functionally interchangeable. You cannot replace one with the other. And second is that the parental genomes have different and sometimes even opposing effects within development. Now, my favorite study actually came about 10 years later in the 1990s, in which chimeric mouse embryos were being generated at this time. So here you can allow development to proceed normally a little bit further into the blastocyst stage, where you have an inner cell mass that goes on to form the embryonic tissues. And you can inject into these blastocysts either by maternal cells or by paternal cells, and then let these embryos develop. And this is still not viable. These embryos will still die, but they make it further in development. And what was observed in these experiments is that the inclusion of bimaternal cells led to embryos that were smaller, whereas bipaternal cells led to embryos that were larger. And my favorite part of this experiment is that there's actually also differences in the growth of the brain. So bimaternal cells actually led to an enhancement of brain growth, whereas bipaternal cells inhibited the growth of the brain. So you have small bodies and large brains, or vice versa. And furthermore, if you look at where these uniparental cells are located in the brain, they're located in different places. So the bimaternal cells are more predominantly observed in the cortex, the striatum, and the hippocampus, whereas bipaternal cells are more predominantly localized to the hypothalamus. Okay, so not only do these parental genomes have different roles in the growth of the embryo, they are already being seen to have different effects on the sort of growth and development of the brain. Now, it wasn't known at this time if these imprint effects are due to some kind of imprinting that takes place across the whole genome, or rather if there are specific regions of the genome that are carrying this parent of origin information. And this was further elaborated upon in translocation experiments, where mice are carrying either reciprocal or Robertsonian translocations, in which they have certain gene regions that are either bimaternal or bipaternal, but the rest of the genome is normal. And from these experiments, it was determined that just a subset of regions of the genome are really carrying this parent of origin information, and certain regions of the genome were identified that have different effects on the growth of the organism, as shown in this example here, or on the behavior of the organism, or on the viability of the organism, like I showed in the previous slides. At the same time that this work was being done in the lab setting, syndromes were being identified in the clinic that were also seen to have parent of origin effects. And I think the most striking of these examples is Angelman syndrome and Prader-Willi syndrome. So Prader-Willi syndrome, individuals with Prader-Willi syndrome are usually most noted for having failure to thrive early in life. And then starting in about childhood, they experience hyperphagia, overeating, and if left unchecked, this will go on to develop into obesity. They also have a number of other abnormalities: cognitive impairment, behavioral problems, hypogonadism, small hands, small feet, and a long list of other symptoms. In contrast to this phenotype, Angelman syndrome patients appear to be entirely different. They're usually most noted for their rather happy disposition. They have a lot of unprovoked laughter and smiling, flapping hands. But again, this is a rather severe disease in which individuals usually have little spoken communication, cognitive impairment. About 80% of patients have seizures. So at first, it seems like these two syndromes are entirely different from one another. The symptoms within patients are entirely different. Yet at a genetic level, they're surprisingly similar. And it seems that these two syndromes are caused by nearly identical mutations within a specific region of human chromosome 15. But it was recognized that actually, if you take into account the parent of origin information, Prader-Willi syndrome is arising from aberrations within the paternally inherited copy of chromosome 15, whereas Angelman syndrome arises due to alterations on the maternal copy. I just wanted to take a brief moment and clarify what I think is frequently a common point of confusion when people begin to sort of think about genomic imprinting. And this is the difference between imprinted genes and imprinted disorders and sex-linked genes or sex-linked disorders. So for sex-linked genes, which perhaps most of us are more familiar with or have heard more about, these are genes that are occurring on the sex chromosomes, the X or the Y chromosome. And therefore, there is a differential sensitivity between males and females to the development of certain sex-linked diseases. Now, this is different than the case of genomic imprinting. Imprinted genes are occurring on autosomes and therefore affect males and females equally. In fact, all of us in this room and on Zoom have the exact same pattern of imprinted gene expression. And imprinted disorders like Angelman syndrome and Prader-Willi syndrome occur equally in males and females. So what's important here is not whether or not you yourself are male or female, but whether or not you inherited a particular gene from your mother versus your father. Okay, so ultimately these early studies led to the identification of individual genes that are imprinted. And by this, I mean that these are genes that are predominantly or exclusively expressed from one of the parental copies. So either a maternally expressed gene shown in red or a paternally expressed gene shown in blue, where the other parental copy is silent. And this is in contrast to your typical pattern of gene expression, where mostly genes are either expressed from both alleles or they're silenced on both alleles. So this is sort of the genome-wide map that exists today for imprinted genes. As I said, these are occurring across all autosomes. There's somewhere in the ballpark of around 150 genes whose imprinted expression has been verified across different studies. So this then allows us to be able to ask a couple of questions. The first is, what are these genes doing? What are the functions of imprinted genes? And second, why do we even have genomic imprinting to begin with? If you think about it, we're diploid organisms, and that gives us many advantages. It helps us buffer against recessive mutations. But for a given imprinted gene, you're effectively haploid. You have only one functional copy of that gene, and therefore we're more susceptible to developing disorders that arise from alterations within these genes. Yeah. So the question was would the mitochondria be an example of an imprinted gene being maternally expressed? I think the mitochondria are a little bit unique in that the mitochondria, right, they contain their own mitochondrial DNA, and mitochondria are maternally inherited. So I've never really thought about it in that way. I guess you could say that maybe mitochondrial RNAs are by default always maternally expressed information. Yeah. Yes. Yeah, sorry, I meant to mention that. Even though most of these studies have predominantly been done in mice, there is a very high conservation of the imprinted expression of genes between mouse and human. So for the most part, all the genes, let's say you have a maternal specific gene in mouse, it's also maternally expressed in humans. Any other questions so far? Okay, great. So we're going to then go on and visit these two questions. What do imprinted genes do? And can we think at all about like why we've possibly evolved genomic imprinting? So the first is that these imprinted genes actually have many common functions. And I'm going to bucket their functions into two main categories. So the first is that many imprinted genes are enriched for roles regulating different aspects of embryonic development, in particular related to the growth of the embryo. So you have imprinted genes that play roles in the placenta, in metabolism, and appetite regulation, et cetera. And in fact, two of the very first imprinted genes that were identified were the insulin growth factor IGF-2 and the IGF-2 receptor. So IGF-2 is a paternally expressed gene. It's acting in the early embryo to help drive growth of the embryo. In contrast, the IGF-2 receptor is maternally expressed, and it's actually acting as a negative regulator of IGF-2 signaling to repress growth. So this is an example of two genes that are functioning in the same pathway, They have opposing parent of origin, and they actually have direct antagonistic effects on one another. And this is consistent with what you see amongst these genes that are regulating growth in the embryo, that paternally expressed genes are typically acting to drive growth, whereas maternally expressed genes are typically acting to repress growth. The second kind of bucket of functions that you see associated with imprinted genes are different roles within the brain. So there are a number of imprinted genes that play roles in processes like memory and cognition, how the organism behaves, vocalization, and suckling. So if we think about perhaps why genomic imprinting may have evolved, there are many hypotheses that have been proposed, but I think probably the most widely recognized is sometimes called the parental conflict hypothesis. This was proposed by David Haig here at Harvard, actually even before the identification of the first imprinted genes. And according to this hypothesis, it posits that the parental genomes within the offspring actually have different interests in terms of the acquisition of resources from the mother, where it's within the interest of the paternal genome to drive the growth of the embryo, whereas it's in the interest of the maternal genome to conserve resources for the mother, so she's able to provide for all of her offspring, including offspring from different fathers. And so a lot of what's seen about the functions of imprinted genes really agree with this hypothesis and are consistent with the hypothesis that paternal genes within the embryo are driving growth, maternal genes are acting to restrict growth. But the exact relevance of this kind of hypothesis toward the imprinted expression that occurs in the brain is much less clear, although it's certainly notable, right, that the brain is a central region of appetite control, even postnatally. So some of these imprinted expression patterns may still be aligned with this kind of hypothesis, but I think right now we just know a lot less about the imprinting that's occurring in the brain, what these genes are doing, and perhaps why it may have arisen. Are there any questions about this? So if we go back now to this sort of genome-wide map of imprinted genes, the next question that I want us to think a little bit more about is, how does imprinted expression arise? How is it regulated and controlled within the cell? If we think about it, in this case, we have two copies of the gene. You can imagine a scenario where the exact same DNA sequence as one another, and they're sitting in the same nucleus, so they're exposed to all of the same transcription factors, or many of the things that we think about regulating gene expression, yet somehow only one of the copies is expressed and the other is silenced. So how does this actually happen? So most of what we know about how the imprinted expression of genes is regulated is informed by studies on these 6 regions of genomic imprinting that I've highlighted here. Okay, and you can see that across all of these regions, you have a number of neighboring genes that are imprinted, and that these clusters of imprinted genes contain both maternally expressed as well as paternally expressed genes. And it was recognized quite early on that all of these genomic clusters contain a region of DNA that has differential DNA methylation between the 2 parental copies. So one of the parental DNA copies is methylated and the other is unmethylated. And this is simply a chemical mark that occurs sort of on top of the DNA sequence. So it's not changing the underlying sequence of the DNA. And therefore, this is consistent with genomic imprinting being an epigenetic phenomenon in which there's no alterations to the underlying DNA sequence itself, but rather there is a chemical modification, and that chemical modification can be inherited. So just to look at this in a little bit more detail, I'll represent methylated DNA as closed circles, unmethylated DNA as open circles, consistent with the nomenclature in the field. Yeah. So the question was, do imprinted genes across an organism's lifetime switch? And maybe I'll come back to that in a moment. I think the typical answer, the classic answer would be no. But in light of more recent studies where we have better ways of identifying imprinted genes and examining what imprinted genes look like over development or between different tissues, it turns out that the answer is yes. Okay, so this is what sort of one of these imprinted clusters generically looks like, where you have a number of imprinted genes, and somewhere within the vicinity of these imprinted genes is a region of DNA that carries differential DNA methylation between the 2 parental copies. And we call these regions imprint control regions, or ICRs. And you can think of these essentially as like the master regulatory control element of the genes within the cluster. And this has been demonstrated in experiments where you can delete the imprint control region, and now you lose imprinted expression of all the genes within the cluster. So genes either become biallelically expressed or they become biallelically silenced. So we know that somehow the imprinted expression of these genes is due to this region of differential DNA methylation, but there are still many unanswered questions for how this happens, particularly the ability of these regions to act sort of at a distance to regulate genes that are further away. And these are questions that my lab is asking that are still under open investigation. Okay, so a lot of studies in genomic imprinting field have really focused on the dynamics of DNA methylation, given that this is really the primary mark that's regulating the imprinted expression of genes. And so I wanted to highlight just some of the key findings from really a large body of work focused at understanding how DNA methylation takes place, how it's maintained, et cetera. So this differential DNA methylation is originally established during gametogenesis. So during the formation of sperm or egg, certain regions of DNA will either be methylated or protected from methylation. And therefore now following fertilization, you can just think of this basically as like a molecular fingerprint that's put onto the genomes to carry forward this parental information. And then following fertilization, now these differences in the DNA methylation marks are highly maintained throughout all of development. So remember, every single time the cell divides and replicates its DNA, it's now going to very faithfully pass on either the methylation or a lack of methylation onto the new daughter strands. So this basically provides a memory to the cell, indicating which parental copy was inherited from which parent. Now, ultimately, these imprint marks must be erased in order for the system to maintain itself, right? So if you are female and you're going to carry both maternal and paternal marks, yet you would only pass on maternal marks to your offspring. So how does this happen? In the primordial germ cells, the precursor of the gametes, those imprint marks are first erased and then reestablished according to the sex of the individual, such that females only pass on maternal marks, males only pass on paternal marks, and therefore the cycle can continue. Now, these dynamics of DNA methylation, and in particular, the ability of the organism to maintain this differential DNA methylation throughout all of development is actually quite remarkable. And it's even more remarkable given the global changes in DNA methylation that typically happen during development. So this is just a simple diagram of global levels of DNA methylation, either during gametogenesis or embryogenesis. And I'll just bring your attention right here, where typically immediately following fertilization, there's a global wave of demethylation that occurs in the early embryo. And then over developmental time in different tissues at different genes, you gain back DNA methylation levels. But for these imprint control regions, which are shown here, you actually maintain these differential DNA methylation marks despite this global sort of loss of DNA methylation across the genome. Okay, I'm just going to take maybe a brief moment here and ask if there are any questions. This has sort of been the historical perspective on genomic imprinting and some of the key findings. I'm going to switch gears a little bit and talk about some current approaches that are being used. Yeah. Let me make sure I understand your question correctly. So I think you're asking if basically an entire chromosome itself is sort of marked as maternal or marked as paternal. So the answer to that is, I think basically no, that rather not the whole chromosome is marked, but there are certain regions of the chromosome that are marked. And those regions are the regions that I sort of indicate here across this genome-wide map. So across large portions of the genome, for example, this segment here of chromosome 1, in which there's no imprinted genes, there's not going to be any mark within that region to say, hey, I'm maternal or hey, I'm paternal.
So basically one copy of all of those. Or will there be a mixture?
So let's say for a given chromosome like chromosome 1, you have one maternal copy of chromosome 1, one paternal copy of chromosome 1. For a given imprinted gene, you can have a gene that's either expressed from the maternal copy or the paternal copy. Sorry, I think I'm not following totally the question. Oh, you answered. Okay, sorry. It might also get clarified here a little bit later too. Are there any other questions? Yeah.
Hi, so we received some questions in the registration. I think this might be a good moment for one big picture question. So Rick Bourne asked, as a systems neuroscientist, I found myself confused by the literature on the prevalence of imprinted genes in the mammalian brain. Initial estimates were in the range of 100 or so, but since then he's heard there might actually be several thousand. What's the current best estimate?
Yeah, so there has been some like kind of mixed report on this over time, where historically through these kind of more laborious studies using translocations, that's where we developed this kind of genome-wide map that I've shown, where there's about 150 imprinted genes or so. Using now modern approaches that I'll talk about in a second, where you can identify imprinted genes basically in a single experiment genome-wide, there were some of these early papers that found a surprisingly large number of imprinted genes. And I think this turns out to be due in part to the sensitivity of the assay and the sort of rigorous statistics that you need in order to call imprinted genes. So I think some of these studies were sort of overestimating the number of imprinted genes, and that largely has not held true over time. Rather, if you look across studies, there are about 150 or so imprinted genes that are commonly seen across different tissues. With that being said, if you think about it, the only way that we know that a gene is imprinted is if a gene is expressed in a particular tissue, right? If it's not expressed, we don't know if it's coming, which allele it might come from. So it has to be expressed. And then you need a way to distinguish maternal versus paternal expression, which typically in mouse experiments, you don't have any way of doing that because you're typically using completely inbred laboratory mouse strains that are completely genetically identical between the two strains. They have no sequence differences. So you need some way that you can distinguish this. And so I think we're still limited somewhat in our ability to even call imprinted genes. We can't do it yet at a single cell level, for example. So it could be that there are certain genes that are only imprinted in very specific cell types that we don't yet know that they're an imprinted gene because we haven't done the right experiment in that cell type to find it yet. So I think there is still some open question in this area, but I think still really the current best estimates are around 150 imprinted genes. Of course, that number will inevitably be wrong, but somewhere in that ballpark. Yeah.
Are there imprinted cancers that contribute partial adjustments?
I would love to know the answer to that question. We don't know. We're trying to do some experiments in the lab to get at that, and maybe I'll sort of hint at this in a moment too. Sorry, I didn't repeat the question for those on Zoom, but it was a question about are there allelic or imprinted enhancers?
One more question that before we switch gears. So Camilla asks, I was wondering if one imprinted gene that is methylated can become unmethylated during the different cycles of oogenesis or spermatogenesis.
Yes. Okay, so let me go back to where we're at, which is here. I'm trying to talk a little bit about the present. So now one sort of genetic trick that people are using to try and examine either DNA or RNA features of imprinted genes utilizes a clever but simple approach in which you can cross two different subspecies of mice, for example, the musculus mouse, and mate it to a castaneous mouse. As I said, these are completely inbred laboratory strains, and we know the complete genetic sequence of each of these mouse subspecies. And this means that now in the resulting F1 progeny, the maternal genome is of a musculus sequence, the paternal genome is of a castaneous sequence, and these two strains of mice, for example, have a single nucleotide polymorphism or a nucleotide difference that occurs about 1 every 150 nucleotides. So you could subject these mice to whatever kind of DNA or RNA sequencing approach that you would like, and use now the differences between the maternal and paternal SNPs to say whether or not a particular DNA or RNA read is maternal or paternal in origin. So for your normal non-imprinted gene, you would have an equal ratio of maternal and paternal SNPs, whereas you could observe maternal or paternal biases if a particular gene is imprinted. So I wanted to highlight just a couple of examples in the way in which these hybrid mouse strains are now being utilized together with modern high throughput sequencing approaches to examine different aspects of genomic imprinting. And the first is to say that we can now get a much higher resolution readout of the DNA methylation dynamics themselves. Remember that DNA methylation has historically been very highly studied within imprinting over time. But we've now advanced to the stage where we can actually read out on single molecules of DNA the methylation versus unmethylation status across long stretches of DNA. And this is in part accomplished using long-read sequencing approaches like nanopore, where you can take DNA, even in the absence of any amplification of that DNA, and by reading it through a nanopore sequencer in which changes in the electrical signal indicate the underlying DNA sequence, as well as chemical modifications on that DNA sequence, you can get this very high resolution readout of DNA methylation. So this is just an example from our lab where we've done nanopore long-read DNA sequencing across a particular known imprint control region indicated here, where now these individual rows are actually single molecules of DNA. The black bars indicate a methylated nucleotide. The red bars indicate an unmethylated nucleotide. And we can use the SNPs within these hybrid cells, these are hybrid neurons, to then say whether or not that DNA was maternal or paternal in its origin. So we've separated these out, and I hope that you can sort of readily see and appreciate that for this imprint control region, it's highly unmethylated on the paternal allele, where it's highly methylated on the maternal allele, and this is consistent with the known sort of methylation pattern for this locus. Second is that many studies are now beginning to examine not just DNA methylation, but other features of the DNA that might be different between the maternal and paternal alleles. And so there's a focus on trying to understand where the DNA is physically located within the nucleus of the cell, and how is the DNA actually folded within the nucleus of the cell, and are there differences in these features between the two parental copies? So this is an example from Fei Chen and Jason Buonrasto's lab here at Harvard, where they performed an approach on these hybrid cells again called in situ genome sequencing, where they can directly perform DNA sequencing in fixed cells and use the SNPs, again, that differ between these two mouse strains to look at maternal DNA versus paternal DNA. And I'm including this example because I think it's very visual, and so it's easy to see what's going on. And so they performed this in single cell zygotes before the two pronuclei had fused together. So you can readily identify the paternal pronucleus versus the maternal pronucleus, and you can actually track where this DNA is going as the cell now divides into a two-cell embryo, where you can see that the paternal and maternal genomes at this stage are still actually quite segregated from each other in the nucleus. Of course, that you can continue to follow this over developmental time and ask other questions about where these parental alleles are actually localizing within the nucleus itself. But as I hinted, there are also studies that are being performed to try to understand how the DNA is actually folded within the cell. And these are using approaches like HiC, a type of chromosome conformation capture assay, where you can, again, get a very high resolution readout of how the DNA is actually folded within 3D space. And this has been done in studies both looking at early embryogenesis as well as across postnatal brain development. And what has been observed from these studies is that actually, if you compare the genome folding between sperm and egg, they're folded very differently from one another. If you imagine, right, the sperm has a very compact nuclei, and the chromatin is very condensed in this state compared to the space available within the egg. And following fertilization, there's a pretty dramatic reprogramming of the structure of the genome. They mostly converge on a common structure, but some differences in the structure remain. And it turns out that at least some of this is due to these regions of genomic imprinting. So my lab is currently trying to investigate what these sort of genome structures look like within imprinted regions. And back to your question, we're very attracted to the model that perhaps there are different enhancer promoter contacts between the two parental alleles that are helping to drive imprinted gene expression. Okay, and then last but not least, of course, we can use this kind of approach to also examine imprinted gene expression itself. And I think that this will maybe get to a couple of questions that were asked earlier. So this is a study performed by Catherine Dulac's lab at Harvard, where again, they have these reciprocal hybrid mouse strains, where they're able to perform RNA sequencing and determine either maternal or paternal expression of genes. And in this particular experiment, they actually dissected out a number of different regions of the brain shown here, compared to imprinted expression of genes across a number of peripheral tissues. And this is a heat map that's showing the allelic bias in the expression of a given gene. So the stronger the red color, the more maternally biased its expression is. And the stronger the blue color, the more paternally biased the expression is. So I hope first what's immediately obvious is that there's a lot more color on the brain half of the slide than on the peripheral tissues, meaning that there's a higher imprinted bias that's occurring for these genes in the brain. And I think that there's been a couple of surprises that have come from these type of studies. And the first is really the dynamic regulation of this imprinted expression across different tissues. So you have genes that are like UBE3A, this is the Angelman syndrome gene. It's biallelically expressed, that's what gray means in these plots, across all peripheral tissues, but it's maternally expressed across different regions of the brain. There's even crazier examples like GRIB10, which is maternally biased across peripheral tissues, paternally biased across the brain. Or you can see differences between different regions of the brain from maternal to biallelic to paternal expression. So this is a little bit inconsistent with historically how we've thought about genomic imprinting, where a lot of that has been shaped by our understanding of the DNA methylation dynamics that's controlling imprinted gene expression. The DNA methylation is the memory to the cell. It's incredibly static. If you look across different tissues, the DNA methylation dynamics typically don't change for these imprinted regions. Yet, the imprinted expression is surprisingly dynamic. And so there's still many open questions about how exactly DNA methylation is controlling imprinted expression and what sort of this diversity of imprinted expression may mean. The second surprise that I think has come from these studies is that historically, we typically think of imprinted genes as being either 100% from one allele or 100% from the other allele. But that turns out for many genes to not be true. Rather, what you mostly see here are imprinted biases, where they're partially expressed from one parental allele, but more strongly expressed from the other. We don't yet know if this is due to a mixture of cell types, where in some cells it's biallelic and other cells it's fully imprinted, or if at a single cell level, you truly have these imprinted biases that exist. So this is still an area of open investigation. I'm gonna give just a quick recap, basically, on what I've told you so far. So genomic imprinting is an epigenetic process. It's regulated by DNA methylation that's differentially inherited from the two parental copies. And ultimately, these differences in DNA methylation lead to genes that are predominantly expressed from one parental allele. These genes have well-established roles in regulating growth in the early embryo, but I think we're now increasingly able to appreciate that they have many roles in regulating different aspects of brain development and behavior. And lastly, that alterations within these imprinted genes are responsible for the development of a number of different imprinted disorders, things like Angelman syndrome and Prader-Willi syndrome. Again, many of these having phenotypes associated with dysregulation of the central nervous system. Are there any questions before I sort of move on from this to work that my lab is doing? Yeah.
I'm curious what's known about how expression from one allele.
Yes, so there are a number of reports, I would say all of them a bit puzzling, where sometimes you manipulate the expression of one imprinted gene and you see changes in like a number of other imprinted genes from other chromosomes. So I think that there are sort of many reports of this kind of randomly in different experiments, but we don't really understand what is going on there. So I don't know, there's definitely no mechanistic insight into any kind of sort of like trans communication between imprinted genes. Yes.
We have another question on Zoom from Sudhir, who asks, are there any instances from other cells or diseases that show that imprinted genes show differential chromatin looping? And if DNA has any role to control looping? Sorry, I'm not sure if I'm reading it correctly.
I think if I understand the question.
Oh, DNA methylation, sorry.
Yeah. Basically, is there differences in the maternal and paternal DNA structure? Looping or TAD formation, maybe is another way of saying it. And the second part of the question, I think, was, does it have to do anything with DNA methylation? So yes, there are differences in the looping between maternal and paternal DNA. This is something that my lab is studying. And the basis of this differences in the looping is actually due to DNA methylation. Remember that that is sort of the basis of genomic imprinting. And so it turns out that one of the key proteins that's involved in forming chromatin loops is called CTCF. This is a DNA binding protein, and it binds to DNA in a methylation sensitive manner, meaning that it's able to bind to unmethylated DNA, but it's not able to bind to methylated DNA. So basically, the differences in DNA methylation can result in differences in the binding of this protein called CTCF, and therefore you can have different loops that form between the two parental copies. One of the questions that we're trying to actively investigate is, does this matter? Does it matter that the maternal DNA is folded differently than the paternal DNA, and what effects does this have? And we think the answer to that question is yes, that the folding does matter, but there haven't been as many studies that have really gotten into sort of the mechanistic impact of these differences. Okay, so the overarching goal of my lab is to try to understand how the genes that we inherit from our mom can have different effects on our brain than the genes that we inherit from our dad. And just to give you a sense of the kinds of questions that we're asking, the first is that we ask a number of questions that are aimed at trying to understand the cellular mechanisms by which imprinted gene expression is controlled. So in other words, how does differential methylation ultimately result in a gene having imprinted expression? And I've given you sort of some hints of the way that we're thinking about this now. The second thing is that we ask questions about what imprinted genes do within the brain, what are their functions? And for this, we predominantly focus on a large number of non-coding RNAs that are imprinted in the brain. So there are long non-coding RNAs as well as small non-coding RNAs that have imprinted expression in the brain. By non-coding RNA, I mean this is a gene that does not encode for a protein. Rather, it functions as an RNA molecule itself. And lastly, we hope to take what we learn about how imprinted expression is controlled and what imprinted genes do to think about therapeutic approaches for different imprinted disorders. So in our last bit of time together, I'm going to tell you about a collaboration that I was involved in that uses information to develop a new therapeutic strategy for treating Angelman syndrome. But before I do that, I want to just take a step back and maybe we can think about general principles that one might like to use to treat imprinted disorders more broadly. So if we just take a given imprinted gene, in this case, we're looking at a gene that's maternally expressed, but paternally silent. This is likely to develop into an imprinted disorder if one were to inherit a deletion specifically from their mother, because in this case now they have no more expression of this particular gene. The paternal copy, however, is perfectly intact. It's a wild type, there's no mutations in it, it's just silenced by genomic imprinting. So if you could perhaps understand how the paternal copy got silenced to begin with, then maybe the ideal sort of situation for a therapy is to take advantage of the fact that it's sitting there dormant in the cell and find a way to reactivate its expression. Now you've taken an individual who would typically have a maternal expression of this gene, you've swapped it out, they have paternal expression of the gene, but the protein that's made is the same at the end of the day, and the cell doesn't care if it's maternally or paternally expressed. So theoretically, this might be able to correct the underlying molecular defects of different imprinted disorders. Of course, the devil's always in the details, and not all imprinted disorders arise from genetic deletions of a given imprinted gene. You can also have epigenetic changes that result in imprinting defects, or you can inherit sort of two copies of the gene from one parent. Now, in this case, if you were to apply a similar approach, you would actually lead to a twofold overexpression of your given gene of interest. So again, sort of depending on the exact genetics that are involved and the function of the gene that you're manipulating, it's important to keep all of these factors in mind. Many imprinted genes are dosage sensitive, so there would be reason to be concerned about a twofold overexpression of a given imprinted gene. So let's talk a little bit more detail about Angelman syndrome itself. This is a neurodevelopmental disorder that I already introduced. It has a prevalence of about 1 in 8,000 individuals. There's no corrective treatment that really gets at the underlying symptoms themselves. Rather, the typical management is done by treating individual symptoms within patients. This is what the genomic locus looks like that's affected in Angelman syndrome. So there's a region of differential DNA methylation, which is the basis of genomic imprinting here. Then actually quite far away from this region, there's a gene called UBE3A. This is a ubiquitin ligase that within neurons is maternally expressed. It turns out that in non-neuronal cells or across all peripheral tissues, UBE3A is not imprinted and it's biallelically expressed. Angelman syndrome is caused by inheriting deletions or mutations in UBE3A on the maternal copy. So this leads to a complete loss of this protein within neurons and a 50% reduction of this protein across other cell types. It turns out that this 50% loss doesn't seem to have that much effect, and most of the symptoms in Angelman syndrome seem to arise from this complete absence of UBE3A in neurons. So we can really try to focus on what's going on in neurons, and really the ultimate idea is to figure out how the paternal copy got shut off to begin with, and then find some way to reactivate its expression. So it was really exciting when in 2012, it was discovered by Art Boudette's lab at Baylor College of Medicine that the paternal copy of UBE3A is actually getting shut off by an imprinted long non-coding RNA. This is called the UBE3A antisense transcript. Again, by non-coding RNA, I just mean this is a gene that's not encoding a protein. And it was demonstrated in their lab that this long non-coding RNA is actually acting to shut off the paternal copy of the UBE3A gene. So this was really great now that we understood, at least in part, how the paternal copy was getting silenced. And it meant that our goal became to find a way to basically repress the repressor. Let's get rid of the long non-coding RNA. And by doing so, we should be able to reactivate this intact paternal copy within patients. So the approach that we chose to use was using an antisense oligonucleotide. And this was a collaboration between Art Boudette's lab and Ionis Pharmaceuticals. And the way that this approach would work is that if you have an antisense oligonucleotide, which is basically a small piece of DNA-like molecules, it's able to bind through sequence complementarity to a target RNA, in this case, this long non-coding RNA that we're focused on. And this hybrid between the ASO and the RNA recruits an endogenous enzyme in the cell called RNase H. RNase H then cleaves the target RNA, leading to its subsequent degradation. And these antisense oligos have been administered in the brain in the clinic. They have a very long half-life due to chemical modifications on the backbone of the ASO itself. So this is basically the idea for this approach: knock down the long non-coding RNA and reactivate the paternal copy of UBE3A. So I'm just going to show a little bit of data to show you what this looks like in the mouse. So these are early experiments that we did using wild-type primary cortical neurons, where we can treat these cortical neurons with increasing doses of the ASO. And this causes a dose-dependent knockdown of the long non-coding RNA and a corresponding dose-dependent increase in the paternal copy of the UBE3A gene. We can also look at the protein levels that occur. In this case, we're using primary neurons from an Angelman syndrome mouse model, which has a deletion of the maternal copy of UBE3A. And therefore, these neurons typically don't express UBE3A protein as seen in the PBS-treated control cells. But if we take these Angelman syndrome neurons and now treat them, these are two different oligos targeting the long non-coding RNA, just showing biological replicates, you can nicely reactivate UBE3A expression within these cells. And you actually in culture achieve levels of UBE3A that are very similar to wild-type neurons. We then administered this ASO into mice. So this is in the Angelman syndrome mouse model, where we administer a single dose of ASO by ICV injection directly into the lateral ventricle of the brain. And this allows the ASO to actually be very widely distributed throughout the mouse brain. 1 month after delivery, we then measured the UBE3A protein levels within mouse brain sections. So here the UBE3A protein staining is in white. The first column is just looking at the normal distribution of UBE3A protein in a wild-type mouse brain. So you can see that there are nuclei of neurons that light up in the cortex, the hippocampus, and the cerebellum. The middle column are from Angelman syndrome mouse model, just treated with a negative control, non-targeting ASO, which mostly lacks UBE3A expression within neurons. But now the administration of this ASO targeting the long non-coding RNA reactivates the intact paternal copy of UBE3A, and we see a partial restoration of the protein within the mouse brain. Of course, the major question is whether or not this actually corrects any of the symptoms in the Angelman syndrome mouse model. And so we took a cohort of adult Angelman syndrome mice, administered 1 single injection of ASO, and then performed a fear conditioning assay 1 month after delivery. So in this assay, you place animals into a particular training environment. You allow them to explore that environment. You can measure their percent freezing. You then administer a noise impulse that co-terminates with a foot shock. And then the next day, you put them back into this training context and again measure their freezing response. So wild-type mice will explore the original environment, but however, after the fear conditioning assay, they'll now freeze in response to being placed within this environment. You can see that the Angelman syndrome mice are defective in this assay. They show less of a freezing response compared to the controls, but the Angelman syndrome mice that were treated 1 month before with this antisense oligo targeting the non-coding RNA now perform similarly to the wild-type animals. So this is one experimental paradigm where we did see an improvement in the mice. However, there were a number of assays that we performed where there was no improvement, including a marble burying test, a rotarod assay, an open field assay, et cetera. So there are still open questions that remain about the timing of treatment, the duration of treatment that would be needed in order for this to have a complete phenotypic benefit. Nevertheless, the publication of this work sparked preclinical and now phase 1 and 2 clinical programs at a number of different pharmaceutical companies shown here. So these clinical trials are ongoing, each using an antisense oligonucleotide in a similar fashion to what I've just described, except of course targeting the human non-coding RNA as opposed to the mouse. As I mentioned, there are still a number of unanswered questions, and particularly regarding the timing of ASO delivery that may be needed. So there are some really nice genetic restoration of UBE3A experiments in the mouse, which indicate that there's a particular developmental time window in which one must restore UBE3A protein in order to correct all of the symptoms within a mouse. Of course, how that exactly translates to the clinic and the timing that would be needed in an Angelman syndrome patient is still an open question. Again, the duration of treatment needs to be investigated. So these antisense oligonucleotides actually have a very long half-life. Again, in the mouse, we observed effects that lasted for 4 months after a single injection. And in large part, the duration of treatment in the clinic is being informed by the successful trials of spinal muscular atrophy by Ionis Pharmaceuticals. There are also other approaches that are now being used to try to perform similar sort of effects on reactivating the paternal copy of UBE3A. So I'm just highlighting one study from Mark Zilka's lab, which came out now just a couple of years ago, in which they used CRISPR-Cas9 to cause sort of genetic aberrations within the paternal copy of the long non-coding RNA. And this actually acts to truncate the expression of the non-coding RNA, once again, thereby reactivating the paternal copy of UBE3A. So there are also alternative approaches that are being used. Just in the last minute, because I think we're about out of time, I wanted to again just zoom back out and talk a little bit about Angelman syndrome versus Prader-Willi syndrome that I alluded to earlier. So we've just gone over more extensively the genetics of Angelman syndrome. This is very clearly due to mutations in the maternal copy of this gene called UBE3A. But what's going on in Prader-Willi syndrome? So the genetics of Prader-Willi syndrome is much more complex. There's likely a number of paternally expressed genes that are involved in the basis of this disorder. But there are a handful of patients that have more minimal deletions, and they occur in this region of the genome here, where it's a portion of this long non-coding RNA segment that we've just been discussing. And it turns out that this region of the genome is actually expressing another type of non-coding RNA called a SNO RNA, or a small nucleolar RNA. We don't yet know what these particular imprinted SNO RNAs are doing in the brain. They're exclusively expressed in the brain. And our lab is performing a lot of active experiments at the moment to try to understand the function of these SNO RNAs as a way of eventually thinking about therapeutic approaches for Prader-Willi syndrome as well. So with that, I'm just gonna conclude. I hopefully told you today about how we can use information about how these genes are regulated and the functions of long non-coding RNAs or other non-coding RNAs to think about therapeutic strategies for imprinted disorders. And then of course, would like to thank all individuals in my lab who help me think about these questions every day. And I would love to take any additional questions that we have time for.
Are there any questions?
Yeah.
So you mentioned earlier in your talk about how so I was wondering if
Yeah, so the question is that for imprinted genes, it turns out that many of them have imprinted biases as opposed to just sort of a complete expression from one allele. And therefore, when we think about treating imprinted disorders, are we affecting just the imprinted biases versus affecting only one copy? So there haven't been a lot of treatments for imprinted disorders that take approaches like this, so I don't know the specific answer to that question. In part, it's because we don't know how these imprinted biases arise. So in the case of UBE3A, it is rather monolithically expressed. There's very little expression from the paternal allele. So for the cases where you have these imprinted biases, I think the first thing is to understand how those imprinted biases arise. And then you would know whether or not your therapy is going to alter both alleles or just one allele, depending on how it works. Right, if there's no more questions on Zoom, yes, no?
We had a question we'd received or two when people registered that we thought were interesting. So in addition to some of the disorders discussed today, what's known about other like just psychiatric disorders, speech and language impairments? Is anything known about the role of gene imprinting there?
Yeah, so there are imprinted genes that play roles in communication. So for example, in Angelman syndrome, these individuals usually have very little spoken communication. They usually say maybe at most 20 words if they're a higher functioning Angelman syndrome individual. Some hypotheses exist, again, sort of related to this parental conflict idea, that perhaps mouse pups that vocalize more are more likely to get nutrients from the mom. So some of these imprinted genes and why they've evolved imprinted expression, even within communication, may be linked to this idea of trying to get nutrient acquisition from the mother. In terms of the other part of the question about imprinted genes in neuropsychiatric disorders, so some of the imprinted disorders do have neuropsychiatric symptoms. You do see some things like obsessive compulsive disorder, anxiety disorders, et cetera. I don't know the answer the other way around. If you had a schizophrenia patient, are they likely to have alterations in imprinted gene expression? I don't know the answer to that question.
So let's see. Yating asks, would imprinted genes act differently in iPSC cells?
So there are some really interesting studies that have been done in human induced pluripotent stem cells, in which you can actually take, let's say, a fibroblast from a patient, you can reprogram it into a human iPS cell, and then you can differentiate it into a neuron. As you can imagine, during this sort of reprogramming and differentiation process, there's a lot of changes in the epigenome. There's a lot of changes in DNA methylation. But again, it's been sort of remarkably shown that you can maintain proper methylation at imprint control regions during reprogramming and differentiation. So you are able to do experiments on imprinting using human iPS cells. And we did, I didn't show the data for it, but we did also do that in the Angelman syndrome project as well, using similar approaches.
That's impressive. So 2 more questions. Nick Bellono asks, how does imprinting contribute to evolution? Are there examples in which new traits are driven by imprinting?
Examples in which what?
New traits are driven by imprinting.
Of course, the tough question would come from my colleague, Nick Bellono. I don't know the answer to that. I mean, I sort of stated what's known about the evolution of genomic imprinting. I will say maybe as it relates to Nick Bellono's lab and the research that they do, genomic imprinting is only observed in placental mammals. So you don't see genomic imprinting in non-placental mammals or in other species. The exception to this is that genomic imprinting independently evolved in plants. And again, this may be related to sort of why genomic imprinting might exist and this idea of the embryo within the mother and the imprinted expression within the placenta being incredibly important for why imprinted genes have evolved.
That's really fascinating. So Sudhir, who had asked the question earlier, says, as you mentioned that DNA methylation and CTCF are important for looping, do we know if during the whole genome loss of methylation during early development disrupts these loops also?
I haven't seen any studies that look specifically at that. I did hint at some of these studies where they've looked at how the structure of the genome changes immediately following fertilization. So looking from the gamete to the single cell zygote, for example, to the 2-4 cell embryo, et cetera. And there what you see really is actually predominantly a global restructuring of the paternal genome, where the structure gets largely lost and then reforms in a similar manner as the structure of the maternal genome. Whether or not these kind of global changes in the structure of those parental genomes has anything to do with the global changes in DNA methylation dynamics, I don't know that that has been specifically investigated. I think we have one question here in the room. Yeah.
I know that in like some scenarios, it's thought that the localization of genes relative to one another within a nuclear architecture can like determine how likely those genes are to be expressed together.
Yep.
And I know that when I talked today that imprinting can change the nuclear architecture. The fact that like imprinting changed the nuclear, like imprinting of one gene changed the nuclear architecture relative to another gene, and that could explain why across chromosomes you can see trans effects.
Yeah, so that hasn't specifically been tested. I think, now with current approaches, you could possibly get at that question. I think what you're basically implying is that there's a chance that maybe imprinted genes themselves, the DNA encoding imprinted genes, might actually be in some kind of like physical proximity within the nucleus, and therefore they're able to regulate one another because they're localized together. That hasn't been shown. There are a couple of examples that have looked at the location of imprinted genes, let's say for a given imprinted gene. Where does the maternal copy of that gene localize versus the paternal copy? And have shown that these two gene copies localize to different locations within the nucleus. And you can imagine, for example, one might be a lamina-associated copy, and the other might be more sort of centrally located within the nucleus. And I think in part the idea there is that the sort of subregion of the nucleus and the environment that it's in may help determine whether or not that parental copy is expressed versus repressed. So I think that's some of the way that we're thinking about it right now. I don't know if there's any evidence, or there's not any evidence yet, that the imprinted genes from like different chromosomes or different regions are actually physically located together in the cell. But really these technologies are like kind of just coming on the stage now. So I think those are things that we could ask.
More questions? So if not, we can wrap it up. So thank you so much, Dr. Whipple.
Thank you.
And I just wanted to mention, if anyone has ideas or requests for other one-on-one topics, you can email us at info@brain.harvard.edu.